To all viewers and listeners, myself Dr. Icha Purak, in front of you to share information about mitosis. The production of two daughter cells from parent cell is known as cell division. All cells arise from pre-existing cells. Cell division is needed for propagation of unicellular organisms, development of multicellular organisms, for growth and healing of wounds, for reproduction of multicellular organisms. The cell divisions are of different types. Binary fission takes place in prokaryotes. Mitosis takes place in somatic cells of eukaryotes. Meiosis takes place in reproductive cells of eukaryotes. Mitosis is known as equational or homotypic division, as two daughter cells formed after mitosis are identical to parent cell in chromosome number. Meiosis is known as reductional or heterotypic division, as chromosome number is reduced to half as that of parent cell in four daughter cells formed after meiosis. Cell division takes place as per cell cycle of organism. Cell cycle has resting and division stages. A cell prepares itself for division by doubling the amount of DNA and other resources during interphase of cell cycle, and it involves G1, S and G2 phases. M phase is the division phase. It consists of karyokinesis or nuclear division and cytokinesis or division of cytoplasm. Time taken by different phases varies with different organisms. Interphase is metabolic and preparatory stage. Nucleus contains nucleolus and chromatin as diffuse tangled mass. Chromosomes are not visible. Nuclear membrane is intact. Interphase passes through G1, S and G2 phases as shown in the diagram. This diagram shows interphase nuclei. This is the nucleolus and this is the chromatin. Now this slide shows eukaryotic cell cycle. M is the mitotic phase. G1 is the gap 1 phase of interphase in which protein, RNA and ATPs are synthesized. S is the synthetic phase. During this phase, DNA replication takes place and the amount of DNA is doubled for the next division to take place. G2 is also preparatory phase, gap 2, and in this mainly ATP is synthesized. M is the mitotic phase. Now today we will talk about mitosis, and special emphasis will be given on plant mitosis. Karyokinesis can be studied under following heads: prophase, metaphase, anaphase, telophase. After mitosis, two genetically identical daughter cells are formed. In plants, mitosis can be studied in meristematic tissue such as root tip or shoot tip, in which cells divide rapidly. Only root tip cells are generally taken to study stages of mitosis to see the chromosome. Stains like acetocarmine and acetouracil are used. The chromosome count for ileum septa is 2n is equal to 16, and this diagram shows LS of onion root tip, but while preparing cytological slides, generally squash method is followed, in which root tip is squashed with the help of taper and thumb. Now the first phase of division is prophase. It is of longest duration. Nucleus during prophase becomes larger in size. In the nucleus, chromatin material appears as thin filamentous uncoiled structure. Condensation of chromatin starts by dehydration. Nuclear membrane and nucleolus start disappearing. Spindle formation starts from two poles on opposite side in plant cell and from two centrioles in animal cell. Here we can see the origin of spindle of present at the two poles, and this is the prophase nuclei. Nucleus is disappearing and nuclear membrane is also degenerating. Each chromosome consists of two longitudinal halves chromatids joined at a point known as centromere, which is surrounded by a protein disc kinetochore to which spindle fiber attach. Now, prophase is followed by metaphase. It is the shortest stage. Chromosome becomes arranged at the equatorial plate of the cell at right angles to spindle axis. Spindle fibers attach to centromere of chromosome. We can see here the chromosomes are arranged at the equatorial plate and spindle fibers are towards the centromere. Chromosomes become short and thick due to condensation. Towards late metaphase, centromere divides into two and each of the chromosome or chromatid or daughter chromosome has its own centromere. Now we can see here this is the chromosome with its two chromatids and there is only one centromere, but this centromere divides into two and ultimately the chromatids separate, each having its own centromere. Now metaphase is followed by anaphase, is rapid and is completed in 2 to 3 minutes only. Each chromatid is pulled slowly towards spindle pole with its centromere. Chromatids appear of different shapes during movement depending upon the position of centromere as V, J or I according to median, submedian and subterminal position of centromere. This happens because spindle fibers attach with the centromere and pull apart the centromere post and the arms trail behind. During anaphasic separation, spindle fibers contract and become shorter. Daughter chromosomes reach towards two poles in the form of two bunches. We can see the bunches in this photograph, the two bunches. Daughter chromosomes reach towards the two poles in the form of two bunches. In early anaphase, bunches of chromosomes are close to each other, and in late anaphase, the bunches are at a distance. Now, anaphase is followed by telophase. It lasts for about 1 hour. The two daughter chromosomes reach two opposite poles. Chromosomes become uncoiled and relax. Visibility of chromosome decreases. Nucleus and nuclear membrane reappear. It is just reverse phase of prophase. Two nuclei are reorganized at each pole, although are shorter in size than that of prophase nucleus. With this, nuclear division is completed and is followed by division of cytoplasm. Now this diagram shows the telophase cell with two nuclei, and in between the two nuclei cell plate formation has started. Cytokinesis. Division is the division of cytoplasm. Division of cytoplasm takes place by invagination from two sides which proceed towards center, forming a cell plate which divides parent cell into two daughter cell. In animal cell, cytoplasm is divided either by furrowing or clustering of microtubules in the center which fuse with each other to form a continuous plate. In plant cell, invagination start from periphery and meet in the center, forming cell plate. After cytokinesis, two small daughter cells enter G1 phase for the next division. Karyokinesis is not always followed by cytokinesis. In such a condition, binucleate cell or multinucleate cells are formed. In plant cell, plane of division can be horizontal, vertical or diagonal. It depends on appearance of band of microtubules in pre-prophase nuclei. Shortening of chromosomes during stages of cell division make their movement easier as compared to very long twisted interphase chromosome. Spindle fibers start disappearing during telophase except in the equatorial region. At this region, they move towards to form a barrel-shaped structure separating the two daughter nuclei. Now this slide shows different stages of division: interphase, prophase, metaphase, anaphase and telophase. And this slide again shows the stages of plant mitosis. This is cytokinesis, where a cell plate is formed between the two daughter nuclei. Now these are some of the photographs. This is of interphase, this is prophase, this is metaphase, in which the chromosomes are arranged on the equatorial plate. This is early anaphase, this is late anaphase. The two bunches are far apart from each other. This is telophase. Now this slide shows animal mitosis stages. Only difference is that in animal, spindle is formed from two centrioles and cell plate is formed by invagination. This slide shows differences in animal cell and plant cell mitosis. This also shows difference in animal and plant cell mitosis. Then significance of mitosis. Mitosis helps in growth of multicellular organisms, helps in healing of wounds, result in daughter cells identical to parent and also each other, giving genetic stability in respect to chromosome number and gene set in the form of alleles. It helps in asexual and vegetative reproduction. Now with the help of whiteboard we can see this is the chromosome, these are the two chromatids, and this is the kinetochore surrounding the centromere to which the spindle fibers attach. These are the spindle fibers, these are the various chromosomes, the various shapes of the chromosome at anaphase, V shape, J shape, I shape, and the equational division. Two daughter cells are formed. Now with this the presentation is completed. Thank you.